Because how do proteins work? Anybody remember this? How do proteins work based on what?
Size. Shape.
Shape, right? And if we keep adding more, will it have the same shape?
No.
No. So since proteins function based on shape, we have to stop when we're done, or we get something that does something completely different. All right, 307. What is a mutation? Amy. I don't think we may not have gotten that to in our notes, but you could read the book.
Well, the change when a mutation is done right, there's a change in the DNA and therefore the protein changes.
That's right. Mutation is when there's a change in a DNA sequence. How do point mutations and frameshift mutations affect protein synthesis, Margaret? What is a point mutation?
I just said that a point mutation who changes single nitrogen base.
That's right. That's right. And what about frameshift?
And a frame. Well, I just said that if they both change like one base, it would change the entire structure.
Well, so a point mutation will only change one amino acid, right? Because we changed one letter. And a frameshift, we'll talk about that today, we lose a letter, so everything shifts over one. And when everything shifts over one, it changes every single word.
Yeah, but doesn't also, if you change a single nitrogen base, that changes the entire structure?
And actually, but it's only going to change one word, right? If I have the word,
so like the entire structure of one word.
yes, but so it's going to change one amino acid, right? Whereas frameshift will change every amino acid after the mutation. In fact, we'll go over that in detail when we get to it in our notes, rather than write it all now. Okay. Why is a mutation of a sperm or egg cell, why would it have different consequences than one in the heart cell? Sarah.
I'm sorry.
Did I skip somebody?
No. I thought John Michael had just gone so. Never mind.
Oh.
Just keep going. Yeah.
Thank you.
Okay. The mutations of sperm and egg cells have consequences that like affect generations to come.
That's right. They can be passed down, right?
And then a mutation of the heart like only affects like the person.
That's
who's
right.
heart.
The individual has the mutation. Very good. What's the relationship between mutations and cancers? John Clark.
Cancer is a mutation of. Wait, isn't that a mutation? Like an oncogene is a mutation?
You're right. So mutations can cause
Cancer.
cancers, right? Technically, they're not the same thing, but a mutation causes a cancer. Why would low levels of mutation be adaptive, whereas high levels might be a disadvantage, Francie?
Well, low levels can be easily disregarded because the other cells take care of it, like
Okay,
you
sure.
said. And then if it's higher levels, it can't be easily fought.
Yeah, sure. That could be. Although there is an exception to this. In things like bacteria, if you have a bacteria and you take a drug that starts killing it, the remaining bacteria will start mutating super fast.
Uh-huh.
Why do you think they might start mutating really fast? Anybody know? Why would they do that?
Yeah.
What are they trying to do?
Infect you.
Well, so you're already infected. Okay,
White
so
blood? No.
let's say you have strep throat, right? And you take amoxicillin, which kills streptococcus bacteria. Why would that bacteria, as it's being killed, start mutating faster? What's it trying to become?
Stronger.
Okay, it's trying to become immune, right? It's hoping that one of those mutations will protect it from the medicines. That makes sense to everybody? Which is why eventually, in fact, the more we use antibiotics, the more risk we are at things. Has everyone heard of MRSA?
Yeah.
Methicillin, Staphylococcus aureus. That's a
Miss
there-
Garner told us about it.
No, was it
It was
Miss
Miss Garner.
It was Miss
Cat
Garner.
had it.
it's a type of staph, and it's not treatable because it's resistant to antibiotics, and it didn't exist until we started using methicillin, which is a major antibiotic. So
For what?
to treat other bacteria. So methicillin used to kill off. So if you had a staph infection, you took methicillin, and knocked it right out. But then somebody took it, and they killed almost all of it, but one of them mutated just right. And so it was immune to methicillin, and it had nobody to compete with because we killed all the other staphylococcus, and so it started spreading all over. In fact, you see it in hospitals a lot. Okay. Any questions on the homework?
Nope.
We have a lot to cover. We're going to go quickly,
What happened in Grey's Anatomy?
but we'll do our best, okay? All right, so we left off in our notes at mutations. And what's a mutation? Did we go over this?
A change in the genetic code.
Change in the genetic code. Now, I think we talked about this at least a little bit. Most mutations are what? Did we go over this? Most mutations are harmful. Some mutations are harmless, and very rarely, but occasionally, mutations can be advantageous. So we talked about light
that mean?
eyes, right? Helpful. Mutations, very rarely mutations can be helpful in a circumstance. If you live somewhere where there's not much light, it's good to be a mutant and have blue eyes, right? Same thing if with skin color. It's good to be a mutant and have light skin if there's not a lot of sunlight, because you need sunlight to actually make vitamin D. So these mutations can be a little helpful. It's good for everyone pretty much to be a mutant and be able to drink milk. We talked about that, didn't we? If you can drink milk, that's because you are in fact a mutant. Most animals cannot once they get past their infancy. Sarah?
Never mind.
Okay, I'll never mind. All right. So the first type of different mutations we want to look at is germ cell versus somatic cell. And I know these are new terms, but they're new terms for things you already know. A germ cell is a gamete. What are gametes?
Sex cells,
That's right. They're cells that, what's their only purpose?
reproduction.
To reproduce, okay? Why would a germ cell mutation be different than a normal cell mutation? In fact, that's actually a question in your homework.
Gets passed on to
That's
generation.
right. So if a mutation happens in an egg or a sperm, it can be in the entire population from then on out, right? Or at least all the descendants of that mutation. But if you have a mutation, for instance, if you have a mole, that's a mutation, right? If you have a mole, that will not be passed down to your offspring, although the tendency to have moles might be. The individual mole itself will not be because it did not happen in an egg or a sperm cell.
But you can have
Okay?
like bad skin that can be passed on, right? Because like my
Right, but
my
moles
mom
aren't
has
really
moles, I don't know.
right. So that's not a bad skin thing. Moles are caused by they're little cancers, right? And so if you have skin cells, melanin cells that are more likely to have tiny little melanomas, which is basically what a mole is. But they're kept in check, right?
Yeah.
If they grow or change, then you go to the doctor, right? That's
Well,
the
I had to
way
go
it's
every 6 months.
Or if you have a lot of them, you just go. I had one on the sole of my foot, and they cut it off.
I had one on the side of my small toe that they cut off.
Yeah,
I
so
had one right here.
oddly enough, if they're on your hands or feet, they're much more dangerous. So if they're on your hands or feet, they'll cut them off.
Look, I had one.
No,
It was like-
I
I
like
don't-
this one. It's
Well, then-
smeared.
Okay.
Look,
then-
I had my ear.
Why the cut it off at first?
That's a mole.
Well, it's a little cancer, right?
It's a freckle.
So and
Some
so
mole or
moles and
freckle.
freckles are different. Moles and freckles are very
I always
different.
thought that was
Okay.
chocolate.
No,
but
it's
for some reason,
smeared.
moles on
I'm
your
busy
hand
this
and
year
moles on
because
your
I
feet
always
because
tell her
that skin
everything she
grows
has
so
some
much
mole and
faster
she
than the
calls
rest
it
of
a
your
freckle.
skin. You can
I'm allowed to
constantly
ask her
vote.
tomorrow.
You know, you ever done this when your hands are dirty and you see those black rolls that end up? That's dead skin around dirt, right? So you have to grow your skin really fast in your hands and your feet. So those are more dangerous
try
moles, and so now they'll just go ahead and cut them off usually.
He said
You know,
after you're
like-
in the dirt. Would
Or
you
now.
personally be able to identify?
Quiet.
Would you personally be able to identify a mole over a freckle?
I mean, not with I mean, it's
Because
obvious.
pretty
when
sure
Busy's
this is a
here,
freckle.
she has this one thing. I'm positive it's a mole, but she always calls it a freckle.
You know she
I
can
need
hear you.
Yeah,
Okay,
and I need you
so-
him to identify it.
The germ cells are going to be passed on. Somatic cell mutations will not. All right, next we're going to talk about a couple different types of mutations. And I'll give you one example of how important one letter can be. There was a professor who graded a young girl's paper in a college. I forget where the college was. I just remember the story. And he meant to write on it, because she hadn't done enough research, and she said she didn't know. He meant to write on the paper, good start, but you must dig. In other words, she must search further in herself and for her work to find something better. But he changed one letter by accident. And I think actually, now I remember, he was typing on it.
Die.
He put "you must die." The woman was emotionally unstable, attempted suicide after
Oh my
this. He
goodness.
lost his job. All kinds of things happened because he accidentally typed or wrote, I can't remember which, one letter wrong.
Why was that an accident?
So have you ever been trying to write one word and then you just see the first part of the word and then you write something else by accident, or you type something else by accident?
Yeah, like if somebody's
Instead of
talking about
plan, you type plant, right?
Yeah.
People do this all the time. He didn't look over it. This is one letter change. A point mutation is when one letter and one letter only changes, okay? It usually does not have a large impact. For instance, if any other letter had changed in this, if you had become an L, would anyone have tried to commit suicide and gotten fired?
No.
No. If he put an I here, would it have been a problem?
No.
No. In fact, of all the possible letters, the only one that resulted in this catastrophic series of events was because he happened to replace just this one letter with just the one letter that drastically changed the meaning.
With point mutations, if like does the one letter that changes have to like change the entire thing?
Okay, usually if there's one change in the letter, it becomes inconsequential. In other words, it just doesn't work, right? It doesn't do anything. It
So-
makes a protein that doesn't work.
If it so there's nothing that one letter changes. There's no time when it changes the whole thing.
There is a time. In fact, there's a few diseases that are caused by point mutation. Most famously, have you ever heard of sickle cell anemia?
Yes.
Sickle cell anemia
Grey's
is
Anatomy.
caused when an A at one point is replaced by a T. And if you just have this one replacement, and really they bond, so it's just a flipping of these letters. If this happens in one gene, you have the wrong shape of hemoglobin, which is what carries oxygen in your blood. And people with sickle cell anemia have a hard time getting enough oxygen. They can't get too excited, they can't exercise, they can't exert themselves because their blood doesn't carry as much oxygen only because of one single letter that got wrong.
Are they the ones that sometimes have to have limbs removed because they can't get enough oxygen to it?
Limbs removed?
Like a foot.
Not too often. Usually they only do that now for like diabetes.
Yeah.
I know it's an off topic, but did the girl survive? Did she live?
From this story, I don't know. I heard it like third hand from a professor.
Did it not make national news?
I think it happened in like
I'm
the
gonna
80s or
Google.
90s. I just had a professor who
I'm
told me that
gonna
story
Google
whenever.
I was just going here and be like, why'd you do this?
Well, good. I'm glad you're not nearly as emotionally fragile as whoever this girl
Mara's
was
not
from whenever
emotional.
the story was. Okay, so it has to be just one that, by the way, if this A in sickle cell, if it changes to anything else, if it's a C or a G, then there's no problem. The cell itself won't function, right? It will not continue on. But if it is this one specific change, it causes a large difference. So generally, point mutations don't have a great impact on the organism, or even sometimes not on the cell. Sometimes it doesn't even ruin the cell. The next one causes a bigger change. In fact, if you'll look in your book, there's actually a decent picture of this on page 300 and that's 306.
Maybe not. Just kidding.
You got it right. Before that, I think.
304.
304, okay. So if you look at the point mutation, if you notice from the normal to the point mutation, when we change one letter, how many amino acids did we change at the bottom?
1.
We just changed glycine to serine. Does that make sense to everybody? Now, to go back to our sentence, you must dig, if we had just taken one letter out, and this is a weird thing about this language, when one letter comes out, everything else moves over, we would get you, nisdig, ig. Right? Everything shifts over here. Okay? So if you look at the picture on page 304, you'll see that a frame shift mutation is when one nucleotide is deleted, and so every nucleotide moves over, which means every single amino acid after the mutation will be changed, or at least affected.
What causes it?
When the DNA is replicating, right? So let's say it's reading A, C, T, G. So as it's going down, binding everything together with T, G, A, what'll happen is as it's connecting, remember how it has to connect these together, bond the base pairs? Well, what sometimes happens is as the zipper is coming, for some reason, let's say this has a broken tip, as it's zipping these together, it'll actually skip that and zip like that.
Ooh.
And so this will fall out.
But the T will stay?
The T will stay, but the next time we copy, we will make a copy off of these three without copying the A.
okay.
Does that make sense?
Uh-huh.
Now this strand will give us a decent one, but that one won't.
Would anything be affected if it like left out the last letter?
The final one would, right? The
Yeah.
final one would be affected. But in a frame shift, we affect everything after the mutation, okay?
So that they all move down one and they're not lined up.
Well, so they all move down one. So if you look on page 304, do you see how when we take out one of those U's, everything moves over? And so now we're reading it the wrong way, right? We're reading the words out of order. The book gives the example, because it's 3 letters, the dog bit the cat. Right, if I take out the T here, I understand almost everything, right? But if I take out the first E, this is the word I get.
The dog bit.
See? And so suddenly everything's changed. Every word's been changed in this sense. And so that's what a frame shift does.
It's like dominoes.
Whereas if it's a point mutation, then only one word changes, right?
It's like dominoes.
It's a bit like dominoes. Everything's
So
affected.
it's like, it has more of an impact.
Frame shift typically has a larger impact than a point mutation, right?
What are some
But
things?
generally, it makes a protein that doesn't function as all. Actually, so the blue-eyed gene's a mutation, right? We talked about that. That's actually probably one of them is a frame shift, right? It's supposed to make a dark protein, but when it throws everything out of order, it makes a protein that does nothing.
So that means there's something wrong with you.
That's right. So light eyes are a mutation, right? We talked about that.
So if that happens to like a stop codon or something, would it? just-
Right. So if the stop codon gets messed up, then the protein will keep getting longer and
But
longer.
what if the one
Does
right
that cause
after it was
cancer?
the letter that?
Was the same letter?
Yeah.
Then it would still stop and you'd be okay.
Okay.
Sometimes a point mutation can happen and cause nothing to go wrong, right? Because if you look at your chart, you'll notice a lot of these things, a lot of codons make the same amino acid. So if you happen to get a codon for the same amino acid right there, everything's fine, nothing changed.
Get it?
Okay, so now we're going to look at what happens when chromosomes change, and we're going to do this quickly. So look on page 306 for easier pictures. Now, chromosome mutations are changes not simply in a single nucleotide, but changes in whole portions of a chromosome. These are large-scale switches, changes that happen within a chromosome. They're going to have in general, they're going to have a much larger impact than the point and frame mutations. There's some exceptions to this, but these will often have much bigger impacts. Just from the name of the first one, in deletion, what do you think is going to happen?
Something's
Something
removed.
deleted.
That's right. A portion of the chromosome will be removed, will be lost, okay? So this results in less genetic material and therefore less proteins. A deletion means that chromosome can create fewer proteins. Now, if deletion is taking some stuff out, what do you think insertion is?
Inserting
More.
stuff in.
It is. Not necessarily new stuff, but this is recopying. Have you ever been copying something down and realized you just copied the same
Yes.
sentence twice?
All the time.
Yeah.
Sometimes the DNA polymerase does the same thing, okay? Sometimes it accidentally copies the same thing twice. So if you look at that picture, what happens to BC?
It gets copied.
It gets copied again. It copies BC once, forgets where it is, then copies it again,
Was
right?
it copying the same chromosome twice?
It will a portion of the same chromosome. So insertion is when a portion of the chromosome gets copied twice. And so you now have a doubling of that genetic material. We have twice as much of the B genes and twice as much of the C genes. And sometimes this can actually be slightly beneficial, and sometimes it's harmful.
What happens to someone when deletion occurs?
Well, it depends what got deleted, right? So if it was something not super important, right? Like if it was something like pigmentation, right? You'd have fewer dark pigments. But if it was the enzyme for, let's say, creating antibodies, right? Then you can't fight disease.
Yeah.
So
what
it
about
entirely depends on what was in that chromosome.
what about insertion?
Well, insertion, same thing. Let's say it creates it's a gene for, say, muscle fibers. Well, you may have some more muscle fibers. But if you have too many muscle fibers, then other things won't form. Let's say it's the gene for a hormone that makes you form bone. Well, you may, if you have two copies, well, maybe that makes you have stronger bones. Or maybe that makes you start getting bone spurs, where bone grows where it shouldn't, which is a really bad thing, right? You don't want bone to grow in the middle of a muscle. It's sharp and it's hard, right?
Does someone with like Down syndrome or like the disjunction stuff that we talked about last section, is like insertion more common with that since they have more chromosomes?
I guess since they actually have one more chromosome, they would have a slightly, very slightly, because percentage-wise, it would be like they'd have 0.3 more percent, or I guess 3% more genetic material, but they would have more mutations just because they'd have more genetic material.
Okay.
Does that make sense? I would expect. Okay, inversion, what happens there?
It's broken off.
They switch places, right? Genes can switch places. This actually seems to happen fairly often. Now, this may not seem like a problem, and sometimes it's not at all, but do you remember how we talked about the introns and how they control whether a gene is turned on and off? Well, let's say that the B gene follows A, which is turned off. So it turns off whatever gene's after it. What happens when it moves somewhere else? It can get turned on. So you may have a gene that's not on suddenly switch places and get turned on just because of that, because it's behind an on switch instead of an off switch. Does that make sense to everybody?
No.
Okay, so
Sorry.
let's say we have, let's say B codes for the famine gene, right? Remember we talked about the famine gene? Storing food is fast, okay? Well, your parents, your grandparents had starved, so your gene isn't turned on. But let's say that first zygote that you were, it has a little mistake where it switches those genes around. And now it's behind a switch that is turned on for something like, let's say, meat enzymes.
Okay.
But now that's going to mean the famine gene, even though it wasn't turned on in your parents, just got turned on in you because it was stuck behind an on switch.
Okay.
Does that make sense? And then finally, translocation is very similar, but in inversion, we switch places on the same chromosome. What happens in translocation?
Different chromosomes.
We move to a different chromosome. Translocations, you would think they happen less common, but they actually do happen on occasion. I don't know if you remember, if you had science with Mrs. Garner, but Catherine had a translocation of extra genetic material, or still has, a translocation of extra genetic material onto her 14th chromosome. So a translocation can give you too much protein at certain times. Although if it happens within a single cell, it's not always necessarily harmful. Sometimes it can be what we call benign. It's harmless if it's in the same number. Okay. So translocations are moving when genetic material moves from one chromosome to another chromosome, as you can see in that picture right there. Okay. Any questions on that? All right. Human heredity. Now, remember we were talking about how pea plants were either tall or short? And you said, but hey, I know some people who are tall and they have short children.
not
Or I know some people who are short children. That's right, they're not pea plants. Human hereditary is much more complex than pea plant hereditary. In fact, it's even way more complex than the complexity we're going to talk about right now. But we're going to talk a little bit about a few factors in human heredity that are a little bit different from simple Mendelian genetics. Although I guess at first we are going to talk about simple Mendelian genetics, because simple inheritance, there are a few traits in humans, not many, a few traits that follow simple Mendelian genetics. 2 alleles, 1 dominant, 1 recessive for each trait. So that's simple inheritance. 2 alleles, 1 dominant, 1 recessive for each trait. This is just like we did with pea plants, right?
Can you say that one more time?
2 alleles, 1 dominant, 1 recessive for each trait, right? Most things in humans are not like this. Height in humans is not like this. Intelligence in humans is not like this. Pigmentation in humans is not quite like this. Very few things are. In fact, some people argue none are. A few things like earlobe attachment and the ability to taste phenylalanine do seem to be like that. There's a flavor that you have a dominant gene for you can taste, and if you don't, you can't. It's not even a very sharp flavor.
What's in it?
Phenylalanine, it's just a slightly sour taste.
I was gonna say, I think whether your armpits sweat a lot or not is.
No, that has to do more with if you're a boy or a girl.
I didn't know.
Yeah, men sweat more than women do.
Yeah, I know, but I thought there
That's
was like
true.
I read somewhere that was like
Oh,
one
yeah,
of the
there
most
is a
simple.
gene.
It's just like one of the most
Yeah,
simple
it's not
genes.
simple, but it's close to it. It's like tongue rolling. It's pretty close to being simple.
Yeah, I didn't know tongue roll was that.
It is. It is to a certain extent. That's right. So if you tongue rolling's dominant, not being unable to roll your tongue is recessive. But there are actually some other things that
Are you able to?
I
You can't do it.
I cannot do that.
Recessive.
You can't do it.
Wait, can I do it?
It's easy.
That's, yeah.
Wait, if you can do it, can you whistle?
No.
Right. So it's not something that they're not trying hard enough, right? It's a bit like saying like, why aren't you tall? Try harder.
Is whistle
I don't
one?
understand
Is
why
whistling?
Is whistling
a
one?
trait?
It seems like, I don't know, like everybody should be able to.
Look, there she is. She's doing it.
They can't, it seems to me like everyone
Look,
should be
look
able
at
to
her
do
try
this.
to do it.
I know they can't.
That's
I
it.
can look
Caesar's
at her. I
wife.
can't do it though.
There's
Let's
no-
do it.
No, wait, is your finger like that
Difference.
too?
Hitchhiker's Thumb.
Wait, the widow's. What in the world?
I've got
Okay.
that.
Wait,
Mine
is
Let's
widow's
stay quiet.
peak?
Let's take a look.
Oh my gosh, this is
Quiet. Let's take a look at a way of following a single trait through a generation. This is something called a pedigree. If you had a pure-blooded dog, you probably know they have to have pedigree. In fact, there's a dog food called pedigree. This is a way of following a trait through a family history, okay? Now, obviously, pedigrees can only be so complex because every individual has how many parents?
2,
How many grandparents?
2,
4,
4. How many great-grandparents?
8,
8,
8. How many great-great-grandparents?
16.
16.
Right, so these numbers get
32.
enormous very, very quickly. So we're going to limit this, but we're going to look at a pedigree right now, and I'll tell you the way pedigrees work. Males are going to be squares, females will be round, and honestly, the reason they did this, and the Victorians came up with it, is because ladies are curvy and men are more upright. That's literally why they picked those shapes. So, round for ladies, square for guys, okay? Now, pedigrees can tell us a lot of interesting things, even without telling us what they're showing. We know in this pedigree there are 2 different traits. One trait is marked by filled-in circles, and one trait is marked by empty circles. Everybody see that?
Yes.
Now, here's the interesting thing. Knowing nothing about this, you can tell me, if you look carefully, which trait is dominant and which trait is recessive. I want you to study this and try to figure out which trait is dominant, which trait is recessive. So just looking at the picture, which one's dominant, Sarah?
The filled in one.
Filled in. How did you know?
Because that's what like the majority of them are. And like,
Well, it's pretty close to half and half,
no,
isn't
but
it?
like, if they were both recessive, if they were recessive, you wouldn't see any.
Okay, so the
Dominant.
parents, so the top line parents on the left, right? They have children of both types.
Yeah, and if
Even though they're the same. So that must be dominant, right?
yeah,
Well, here's another question. What must be the genotypes of the parents on the top left?
heterozygous.
They have to both be heterozygous, right? Well, now you can figure something else out. The parents on the right, what must their genotypes be?
One's heterozygous.
Heterozygous.
Well, so the dad's heterozygous, right? What's the mom? Homozygous, recessive, right? You figured all that out. In fact, we can go down and figure out who is who as well. For instance, that square in the middle, the light square, we know what's its genotype?
Homozygous.
Homozygous recessive. And his wife, what must she be?
Heterozygous.
How'd you know?
Because they have a.
Because they're children, right? So we don't even know what that trait is talking about, and we can already read it. Do you see how this
We're so
works?
good at this.
You're so good at that. Pedigrees can tell us what's dominant, what's not. In fact, you can do a pedigree, if everybody remembers the Habsburgs. Everybody remembers the poor Habsburgs and their weird
Ugly.
mutations. The Habsburg chin.
What
you
is
know,
that?
Oh
Big
yeah, the
chin.
big, like a plate.
No, the plate was the collar he wore. So that
so funny.
I know in history,
What page is
important
that?
part of the Holy Roman Empire,
Yeah.
319.
How'd you know?
So the Habsburgs are going to have, you can actually follow a Habsburgs line and find that kind of jutting lower lip, or really underbite as it's called. They have a severe underbite. You can track the underbite going through. You can actually look at the Russian royal family and track the hemophilia that went through the family. Do you know what hemophilia is? It's where you can't stop, your blood doesn't
My
clot.
cousin has that.
So back then, hemophilia was almost always lethal because they had no way of adding clotting factors. But now they can do it without too much difficulty. Okay, so pedigrees, you need to be able to read a pedigree, be able to follow it. I think we did a pretty good job of that.
We did.
So now I want to look at, what's next, thank you, simple recessive traits. Simple recessive traits
cystic
are
fibrosis.
traits that only show up if the individual is homozygous. They only show up if the individual is homozygous. So heterozygous individuals don't have it, which means they can be what we call carriers. Carriers are people who carry a recessive gene but do not show the recessive trait, okay? They carry the recessive gene, but they do not show the recessive trait. Yes, ma'am?
Is that why Miss Garner was like, I have to get tested to see if I'm a carrier of MRSA? Because she told us that.
Well, no, MRSA is not a genetic condition.
Would they carry the trait but don't display it?
That's so right. So that's what a
And those are
carrier is.
heterozygous, you said?
Because they're heterozygous and the trait's recessive.
Oh, I thought
Okay?
they only show up if the system's homozygous.
The trait only shows up, right? So people who are heterozygous don't have the trait.
Oh.
Wait,
Now,
Well, they do.
one of the
but they
most
show up.
common diseases that is simple recessive disease is cystic fibrosis. And cystic fibrosis due to a bad protein for something called a chlorine channel. Not enough fluid, not enough water leaves the throat and the lungs to get to the mucus, and the mucus stays very dry and very thick. And what this does over time is it gradually blocks the air passageways and almost exhausts to death the individual who has to forcibly expel and cough and get that dark black mucus out of their lungs. Until about 30 years ago, the life expectancy for cystic, I guess it's 50 years ago, the life expectancy for cystic fibrosis was 7 years. Okay, so this killed people when they were children. Now the life expectancy, I think, is up to like 36, maybe up to 40 now.
Yeah, I
Wait,
read
so
an
it's
article
like-
about this runner girl that had it, and she ran to like get out of her lungs or something.
That's so now they have all kinds of saltwater treatments, or that parents will clap the children on the back to knock it loose. So it's very treatable now. Very soon it may be life expectancy may be up to 40 or 50, but they've made huge advances in cystic fibrosis. But it is simple recessive, and the reason a lethal disease like cystic fibrosis has stuck around is because it is recessive. I want you to think about this. If you had a gene that killed you when you were 7, what are the chances of it getting passed on?
It's not, because you're not gonna.
You're not, that's right. But since heterozygous people can pass it down without the trait showing up, this gene can stay hidden and be passed on until 2 heterozygous parents have a homozygous child.
What did you say simple recessive was?
Simple recessive means the trait is only expressed if it's homozygous, which means people can be carriers for that trait, which means they have the gene but not the trait, right?
I have 2 questions. Is there a way to like find out if you're like homozygous or heterozygous?
Well, if you're homozygous, then you have cystic fibrosis. The only way to find out if you're heterozygous is to have a DNA test.
Okay, and also, is like Alzheimer's like a thing like this? That's like.
No, it's more complicated. It seems to be more environmental than genetic,
Okay.
but it is partially genetic too. It's more complicated. So all the traits we're not talking about that humans have are more complicated than this.
Is like breast cancer like hereditary?
A little bit. It seems to be more environmental as well.
Okay.
What do you mean by environmental?
It's caused by what happens to you during your life rather than the genetics, your individual gene.
Yeah, but what do you do in your life to cause Alzheimer's?
Well, it's caused by deterioration of proteins in your brain, right? Which can be caused by a decrease in activity. For instance, people like older people when they retire, Alzheimer's seems to accelerate like crazy. People who don't retire actually are very good at fighting off Alzheimer's. It seems to be sedentary lifestyles to contribute rapidly to the development of Alzheimer's.
I gotcha.
When you have Alzheimer's, don't you normally die from like something else with that, or like? Okay.
My grandmother died from Alzheimer's.
My grandfather did too. You're thinking of HIV, AIDS. I think.
You're right.
AIDS, it kills your immune system, and then something else kills you.
Okay.
So, a friend of ours the other day told us that, like, using a certain kind of antiperspirant or deodorant with the aluminum in it causes, but I read up on it, and it said that it didn't, because I was kind of freaking out just a little bit. I thought I was about to have Alzheimer's.
Yeah. So people,
I'm sorry.
so the rumors about what causes the serious diseases that we face are rampant and everywhere. Like you'll find people who tell you that bread causes Alzheimer's, right? Because these are people who are like paleo people, or people who are anti-gluten. Will tell you gluten causes Alzheimer's. There's not a simple, not that it's, I mean, there are celiac people who can't take gluten, but there are other people who aren't, who don't eat gluten because they think it causes Alzheimer's. That was my point.
People tell me caffeine's gonna give me cancer.
Life gives you cancer. Right, so they're not,
saying.
no, there's no
It
study
looks
that
like your
says
ears are
that.
pierced. Gracie, look at this. It looks like his ears are pierced right there.
My ears
Gracie,
pierced.
look.
So they've
It looks
never,
like
okay,
his
so
ears pierced.
there's never a definite.
It kind of does.
Right? All they can show is there's no evidence, right?
Don't touch her.
Hey,
What?
hey,
Don't touch her.
Because some of
That looks cool though.
She's special.
Look, it's
and
my
then
no
the-
one reads the
My
research
hiker's
except
thumb.
for a small group of people.
By me?
Right. Everybody else hears something and goes, "Oh, did you hear?"
Wait.
And because the research for most people is not fun to read, but yeah, anytime you hear a claim like that, by all means look it up. If I make a claim like that, by all means look it up.
I looked it up.
Okay.
If you make
So
a claim
we
that
have the cystic fibrosis, simple recessive. much, much, much rarer. In fact, I only know of one actual dominant, simple dominant disease. Simple dominant means the trait is expressed if there is any dominant gene. The trait is expressed whether it's homozygous dominant or heterozygous dominant. Now, this means whoever has the gene has the trait. Why would a lethal disease that was dominant not get passed down?
Because they died before they could.
That's right, it would kill them. The one exception to this is a horrible disease called Huntington's disease.
I've heard of that.
Huntington's disease typically doesn't set in until about 30 to 35. It's always lethal. It kills 30 to 35, and what happens is a malfunctioning protein has built up in the brain, and the individual begins to go insane, sometimes violently insane, and then they die generally within 5 years of onset of Huntington's.
Due to a protein in the brain?
That's what causes Alzheimer's. A bad protein in the
Is
brain.
it like a mutation?
It's a broken protein that doesn't have the right shape, and it builds up.
Okay.
And because it's a mutation, dominant mutation. And if anyone has this, now the problem is that this is it's completely untreatable. If you know you have Huntington's, you know sometime in your early 30s you're going to start to go insane and then die from it. And the fact of the matter is because it's dominant, if you have it, if you're heterozygous for it, if one of your parents had it and you got it, there's a 50-50 chance you'll give it to any children you have. Children who, after getting tested for Huntington's, would know that when they turn 30, they would go insane and die, right?
Well, then just don't have kids.
Well, that's a question. A lot of people will actually opt for in vitro fertilization.
But
They'll
there's a
actually
50-50
fertilize eggs
chance.
and then check them before they implant
There's
them
a
in there.
50-50 chance.
There's
So you might
you're
not
right.
have it.
You're right. I mean, it's a choice you have to make. The suicide rate for Huntington's is incredibly high, right? When these people who know it's coming, when they feel themselves starting to slip, there's very, very high suicide rate. They just don't want to go through the
Is
process
that like
they
wrong
know
though?
to do.
Because like they're gonna die anyways.
Is it like-
Well, I mean, that's the question. It
Isn't
depends
there
how
a
you feel
thing
about
where you can
suicide.
get doctors? All right, they like.
Well, it's illegal. Doctor-assisted suicide is illegal.
Kevorkian
But
did
a lot
it.
of people think it's morally right.
Like in
Wait,
what
what if you
state?
ask-
Like,
No, the doctor cannot help you commit suicide without committing a crime. Right.
Not even if like you
Dr.
Didn't they do it
Kevorkian did it back in the
somewhere
1920s.
There are a few. Okay, so there was a woman who actually committed suicide recently because she had a terminal disease, but the doctors did not assist her in the process, but she did obtain the chemicals necessary for it, right? But the doctors did not assist. So doctor-assisted suicide. Doctors do not have to stop a suicide by law, but they cannot assist right now. Although actually Oregon did pass a right to die law where doctors can, well, to a certain extent, they can provide materials as long as they are not the ones initiating the process. And I think Oregon's the only state with a right to die law,
That's
though.
awful.
So like, if a person was in the hospital and they didn't have any other family except for the doctor, like that was the only family had left, and it was up to the family to decide whether to, like.
Okay, so that's different from pulling the plug, right? So assisted suicide means an active act to kill the patient. You can, and doctors can do this, and patients can request to withdraw life support. That's a different thing,
Withdraw
right?
care.
Withdrawing life support means no longer forcing the body to stay alive, so you're allowing the body to die. Assisted suicide means causing the body to die. In other words, injecting something, doing something that causes death.
Stupid Adam and Eve.
Yeah. It's useful. I'm not kidding. It wasn't useful. I was joking. I was joking. It wasn't
It's
useful.
the devil.
It's the devil. That old snake. Okay. So, we have this Huntington's disease. Now, by the way, why has this been passed on? Since it's a lethal dominant
Because
disease.
you don't find out until you're like.
That's right. It doesn't take effect till after reproductive age, right? So it can be passed down because it doesn't take effect till after reproduction. Okay. So let's take a look at the last thing, a little more complex inheritance. Complex inheritance is where we don't have just two alleles, one is dominant, one is recessive. It's not simple Mendelian. There's more stuff going on. Now, if you'll remember, when we talked about how Mendel's idea worked and how smart he was, we said that the blending theory, everybody remember the blending theory of inheritance? We said, of course, it's wrong, and that's why Mendel's right. Well, it's not quite that simple. Sometimes the blending theory actually works. It does describe a few
What's the
things.
blending theory?
The idea that if you have tall dad, short mom, you get medium, right? Everything blends.
Can that not happen?
It's not a law like that, right?
Uh.
It's not always how things happen. Height is so variable. It has a lot to do with how well you're fed when you're a kid.
And doesn't it, like, there's not like a standard for tall and short? Like,
Well, I mean,
right?
just compared to everybody else, right?
Oh, okay.
So 5'10", 40 years ago was tall for guys. Now it's
For
average.
guys?
Uh huh.
Oh.
What you're saying,
Um,
girls, and I was like,
My mom's
So-
like 5'11", I think, 5'10". So,
Okay.
like, is that considered tall?
For females, yes.
that's 6'6".
So yes, Amy.
Nuh-uh.
What is it doing?
He's 6'6". Dang.
I mean, just that.
No, I know, so like,
My uncle had an exchange student that stayed with them.
Hey, I mean, it's not. That's not even true.
Can
about
he
me,
walk
right?
through the
You know,
door?
people say, look how big his feet are, really big.
My grandma's
That's not even always
not,
true,
but she's
though.
still on the couch.
So it's not like a law. Sorry.
yeah.
Okay. So we have this complex inheritance, and we're going to look at something called incomplete dominance. Incomplete dominance means that the heterozygous genotype has a unique phenotype. The heterozygous genotype has a unique phenotype. I know those are fancy words, but let's look at what it means. In our picture, we have a red snapdragon, and we have a white snapdragon. Which one looks to be dominant in the picture?
White.
Well, okay, which one gets the big letters?
Red.
So let's look again. Which one gets the big letters?
Pink.
Oh, the red.
The red. The red gets the big
What I
letters.
said.
I know. I want everyone else to say it.
Oh, sorry.
Okay. Now, if this was simple dominance, all their offspring would be what? Red, right? Because we know all their offspring will be heterozygous. But if you read, what color are they?
White.
Pink.
They're pink.
Pink.
So read what the offspring. All the offspring are pink. Because they blended. Sometimes this is what happens. Human eye color at a certain level is incomplete dominance. If you have a bunch of brown eye genes, you have dark eyes. If you have no brown eye genes, you have light blue eyes. But if you have about half brown eye genes and about half non-brown eye genes, you get kind of light hazel eyes, right? Light brown eyes, green eyes. All these come from a kind of blending that takes place. In fact, if you know, it's once people get to be about your age and their color comes in, right? Babies have blue eyes, but about your age, there aren't that many blue-eyed people. Most people have green or brown eyes, right?
I looked something up and it said that green
Was what?
was the most rare. I was looking for
So
it was like brown was like 95%.
So most people have brown eyes, but by brown they usually mean the range of brown to green, like hazel eyes, light brown eyes, right? So those get called. All those are blends, though. You know, there are people who have like almost black eyes, right? Their eyes are very, very dark, very, very dark eyes. And there are people who have brown eyes that are a lighter brown. Sure, lighter brown eyes. There you go. So there's a wide range because of this incomplete dominance. Okay, there's another type of complex inheritance called codominance. Codominance means there's more than one dominant allele. There's more than one dominant allele. Now, the most obvious case for this in humans is in blood types. Does anyone know their blood type?
No. I looked at my birth certificate, it's not on there.
Okay, does anyone know what the options are for blood
Oh.
type?
A positive.
AB.
A,
positive.
B, and A. We're ignoring positive and negative right now. Right, everybody's heard of that?
Yeah.
A, B, and O are the types of blood. Now, if you have somebody who is type A blood, and they have, let's say, a father who's type O blood, all the children will be type A, because A is dominant. Okay?
Wait, why is there two different letters? I thought they had to be the same letter. I thought you used
Not
the
in
same letter twice.
codominance, right?
Oh,
We're in
yeah.
complex inheritance. Things
Oh,
have
blessed.
changed.
Which one's the universal dominant?
Okay, we'll get to that in 1 second, I promise. Okay, so A is dominant. Same thing happens if we have type B crossed with O, because O is recessive. In fact, O is not an O, it's a 0.
BO.
Okay? That's right, it's a BO body type. All right, so O is a 0. Remember how we talked about name tags on cells?
Yes.
Well, there are two different type of blood name tags. There's type A tag and type B tag. So if you have type A blood, what name tags do you have? Type B. If you have AB, A and B. If you have type O, 0. You have no name tags, okay? Remember, the name tags identified it as belonging to you, so your body didn't attack it. So if you have type A blood and you get B blood, what's your body going to do to that blood? Attack it, right? Which is bad for you. But if you have type AB blood and you take B blood, what'll happen? Because
They'll
that's an
accept
okay
it.
name tag, right?
Yes.
In fact, does this have any name tags?
No.
So who'll take it?
Everybody.
Everybody, right? And AB can take blood from everybody. Does that make sense?
I want to be a B.
But if you're O, can you take A blood?
No.
B blood?
No.
AB blood? You have to have type.
But you're a universal
Oh.
donor, though, right?
You're a universal donor.
best one to
This
be
is
is
the
AB.
universal acceptor.
O positive,
Oh, negative.
but negative.
Yeah.
Hey,
But we're not worried about
didn't
that.
finish.
You're right.
Press the
Homework
button.
tonight.
You wanna press the
Homework
button?
tonight.
Sure.
Page 311, 1 through 20.